Okay, so today I'm going to talk about new breeding technologies that may be able to help us achieve durable resistance. So just to get back to the definition of durable resistance, since this is one of the last talks of the session, just to give us a reminder, that durable resistance is something that we can only really tell in retrospect. It's a resistance that remains effective during its prolonged and widespread use in an environment favorable to the disease. So we never really can tell in advance. But with that said, in the case of rust and not only rust but other specialist fungal and bacterial pathogens, it's been observed that quantitative resistance tends to be more durable than single genes. And in rust, we know that slow rusting and adult plant resistance is often quantitative. So if you're not used to hearing about the term quantitative resistance, generally you can think of adult plant resistance as being quantitative. And here's just a graph showing how just 3 loci can give you a quantitative kind of variation. So you might think, well, rust is not as quantitative as yield, but still you can see quantitative variation. And like we saw with Matt Rouse's presentation, the quantitative resistance also shows interaction with the environment, which is something very typical of quantitative traits. So in theory, it's been suggested that pathogen populations can evolve to slowly overcome quantitative resistance. However, this would be a slow process and not a boom and bust type of process, and it would be difficult to detect. So yes, no resistance can last forever, but this type of resistance can hopefully remain more effective for a longer period and avoid that boom and bust. So to breed for quantitative resistance, we're really talking about recurrent cycles of breeding. It requires a screening nursery, existing genetic variation, and it may or may not include the use of markers to select for known loci. There are a number of new breeding technologies. We just heard a lot about speed breeding, so I won't talk about that. Genomic selection is one. This enables selection before lines are grown in the field or when phenotypes are not meaningful. So this would be in an environment where you cannot screen for your trait of interest. That's what I mean by not meaningful. And high throughput phenotyping is another technology that can enable selection before phenotyping for the trait of interest. So this would mean that we can phenotype for other traits, indirect type of traits, but we cannot actually phenotype for the trait we're interested in. So that's where I'm going to focus on the next part of the talk. So what exactly is genomic selection? How do we do it? With genomic selection, it requires that we have a training population that has phenotypic data and selection candidates without meaningful phenotypic data. And both of those sets of lines have markers, and if we have pedigree, we can also use those as well. And all the data is put into a prediction model, and then we have predicted breeding values for all the lines, including the training set and the selection candidates. In this way, we actually get predictions for selection candidates that we don't have our phenotype for the trait of interest. So what's the key applications of GS and what's the advantages? One is we can reduce the time required in the breeding cycle by being able to select new parents earlier. So instead of waiting until we do advanced testing, we can select parents a little bit before. And another possible advantage is we can increase the accuracy of selection. And that is especially the case when we use genomic predictions in addition to phenotypes for the trait of interest. So it's not necessarily only selecting based on prediction, but we can use it in addition to improve our accuracy. So I'll mostly be talking about the first application, which is reducing the time. And the basic idea here is that we can reduce the breeding cycle time. We most likely will have a reduction in accuracy because we're not selecting upon the actual trait of interest. So it's a type of indirect selection. And you generally always have a slightly less accurate selection. However, if you can reduce the time more than you can then you reduce the accuracy, you still come out with having genomic selection more, leading to greater genetic gain than phenotypic selection. If you reduce the accuracy too much, then you will actually not have greater genetic gain. So that's really the key to increasing genetic gain with this type of genomic selection approach. Now, we actually did an experiment where we compared phenotypic and genomic selection in parallel, side by side, in breeding for quantitative resistance to stem rust. In this experiment, the key objectives were to compare realized and expected genetic gain from between genomic selection and phenotypic selection using equal selection intensities. So we really did the exact same type of selection in each case. We also looked and compared if the realized and expected genetic gain were in agreement. So this is just an outline of the breeding schemes that we implemented. And we started out with a base population, which is called cycle 0. Everything was replicated twice, so we did the selection experiment 2 times. And with genomic selection, we were able to do 2 cycles of selection because we could do it in half of the time, whereas on phenotypic selection, we did 1 cycle. For the training sets that we used, were different in the 2 different cycles of genomic selection. In the first cycle, we had a set of 365 historical individuals. Now, this isn't historical in the sense that they're really old, but it's just data that's already pre-existing that we used for the purpose of training the model. And these were 365 lines evaluated across approximately 4 environments each line. And these were in Kenya and in Ethiopia. So this was stem rust race UG99, or the race group. And in the second cycle, we actually used the same set, but then we were able to update the model with the previous cycle data. So we had the 365 lines plus 504 lines that were cycle 0 individuals. And these were evaluated across 2 environments in Kenya. The cycle 1 accuracy, as you can see here, was much lower than cycle 2, and that's something which is pretty expected because the cycle, when we updated the model in cycle 2, we were actually using very closely related lines for the model training. So the cycle 1 accuracy is about 0.3, which is fairly low, and almost to 0.7 in cycle 2. And for the phenotypic selection side, we used unreplicated data from 2 seasons in Joro, Kenya. We also incorporated the pedigree, and our accuracy was extremely high. And this is because stem rust, adult plant resistance, is a very highly heritable trait. So our accuracy was 0.82. Now, we calculated the expected genetic gain in a retrospective way. We used the populations themselves to estimate the parameters. So we couldn't actually predict genetic gain in advance, but we could estimate theoretically what we should be expecting. And we used the theoretical gain from selection formula, which includes the selection intensity. There's another variable of the covariance between the selection unit and the individual in the improved population. That's actually a function of how inbred the lines are. So the more inbred the lines are, the greater genetic gain you'll have. And then there's the additive genetic standard deviation and the selection accuracy. So we took the populations that we developed from selection, and we had cycle 0, cycle 1 of phenotypic selection, and cycle 2 of genomic selection. And in the phenotypic and genomic selection populations, we actually had 2 replicates of each. So we had the 2 replicates of all the populations. So in total, there were actually eight total populations. And they were all evaluated in Joro, Kenya, and Debrecen, Ethiopia in the off-season of 2014. We also looked at pseudo-black chaff in the populations, and that was evaluated in Joro in same environment, 2014 off-season. Now, the realized gain trial, we got really lucky, and everything turned out really well. The heritabilities in Joro in that season were 0.79. In Deborah-site, they were a little bit lower, but still very high, 0.62. And the entry mean heritability across environments was 0.78. We didn't observe much G by E here. As you can see, we just are plotting the population means across the different environments, and you can see clearly that Deborah-site has higher disease pressure, but we did not observe crossover interactions, which is a very, that means it's, the lines that we identify to be resistant in one site are also resistant in the other site. So we look at, this is just plotting out the realized and expected gain from selection, and the blue lines are phenotypic selection, and the red lines are genomic selection. As you can see, the stem rust severity is decreasing over time. So if you look at the A and, well, the A plot is really the most important because it's the realized genetic gain for stem rust severity. And you see that they're very similar in the trend. The expected is also very similar to the realized, which is very good. You know, we're basically agreeing with theory, so we're confirming what we see. And then, interestingly, pseudo-black chaff, we also see a significant correlated response. Now, overall, we could not detect a significant difference between gain from selection in genomic and gain from selection in phenotypic selection. So that's just telling us that in this particular experiment, we saw the exact same genetic gain per unit time from GS and phenotypic selection. So the key lessons that we learned here is that, well, in our particular case, GS did not outperform phenotypic selection. In order for that to have occurred, we would have to have higher prediction accuracy, or if the genomic selection was cheaper, then it would still be favorable. So given that the genetic gain were the same, if you have a cheaper system, then you're still coming out ahead. But in our case, genomic selection was quite expensive compared to phenotypic selection, and we did not actually achieve high rates of gain. Now, knowing that we know now, with the very first cycle being very low accuracy, what we should do in the future would actually be to do the first cycle of selection based on phenotype and the second cycle of selection based on GS, but we can't go back and change it now. But that's another lesson that we learned. So we start thinking, how do we achieve higher prediction accuracies, and how do we reduce the cost? Reducing the cost is really the main issue that prevents us from really using GS in practice. So we have now the ability to use high throughput phenotyping. We haven't really gotten too far into using it for predicting or selecting for disease resistance, but we're pretty far ahead in using high throughput phenotyping to collect data on various traits that are associated with yield. So in high throughput phenotyping, we're basically able to generate thousands of variables useful for prediction modeling, and it really hasn't been used very much for prediction modeling in the past, but it's something that we really should be using it more and more for. One of the huge major advantages of this is that generating high throughput phenotypic data is about 20 times cheaper than genome-wide genotyping at the moment. And by using this data, we can potentially increase prediction accuracy. So this is just a concept of how we can be using high throughput phenotypic data for prediction, and it looks very similar to how we would use it for GS. We have a training population, and we have selection candidates. The training set has phenotypic data for our traits of interest, whereas the selection candidates do not. But all of the lines, both the training and the selection candidates, have data for high throughput phenotypic data. They may also have marker data, and in our case, we also have pedigree. We can use all that data in a slightly different kind of prediction model, but still very similar, which is a multivariate or multi-kernel prediction model. And then we can get predicted breeding values for the traits of interest. So just to give you a little bit more information about the prediction models, in GS we have an ordinary kind of mixed model where we have the phenotype is Y. The first term is fixed effects, and the second term are random effects for the genotypes. And then we use a relationship matrix, K, to model the covariance among the genotypes. In the multivariate version of this, we just simply add more phenotypes. And so this can be the correlated phenotypes from high-throughput phenotyping. Another way we can use the data is to generate a relationship matrix using high-throughput phenotypic data. So basically replacing the K, replacing a marker relationship matrix with a phenotypic relationship matrix. And we can use multiple relationship matrices to incorporate both kinds of data. Now, the stage where we are really interested in applying the predictions is in this stage where after we're done fixing the line, and we are at about the seed increase stage. So we already have to grow the lines in the field anyways, because we need to increase the seed. And at that point, we might have between 30 and 50,000 lines. Now, just to give you an idea, to genotype at the current cost of genotyping 50,000 lines costs $2 million. So it's clearly cost prohibitive to do genotyping at that many lines. So this is why we're really interested in getting the cost down. So here are just some results from predictions that we've done using high-throughput phenotypic data in addition to either pedigree or marker data for prediction, and this is for yield. So you can see ordinary pedigree predictions are in the dark blue, ordinary genomic predictions are in the lighter blue, and then we have dark green is pedigree plus high-throughput phenotyping, and lighter green is genomic plus high-throughput phenotyping. So you can see that having and this is in optimal and late heat conditions. So you can see having the high-throughput data really improves accuracy. And another interesting aspect is pedigree plus high-throughput data is more accurate than ordinary genomic selection. So here we have increased accuracy and 20 times cheaper cost. Now that's for yield, but this is a conference about rust. So can we use this for rust? And we're just starting to get into this. Graduate student Margaret Krause is working on trying to use high-throughput data for multiple traits and not just yield, including different disease traits. So in this case, we're using the Spectre data in the multi-kernel approach, so using the data to generate relationship matrix, a phenotypic relationship matrix. And this is data collected in the absence of any disease. This is just data from optimal growing conditions. And the variables collected, this is collected through an aerial platform, and they're used to generate a relationship matrix for mixed modeling. So here is just a little bit of progress so far towards, and we're just scratching the surface on this, so it really is in early stages and we have a lot further to go. But here's just a relationship matrix generated using Spectre data from one disease-free environment, and that's the information we're using for prediction. And then on this table, I'm showing the accuracy of prediction for stem rust and using different kinds of data. So one is pedigree and markers, and it's about 0.7, a little higher. And then we have spectra from one environment, it's about 0.2, and spectra from 5 environments, about 0.4. So we're not quite there yet to the point with yield, but this is still really interesting because we don't even have disease in our environment. So this is just showing that it is just prove the concept that it's possible to use this kind of data also for traits that are not yield. So just some conclusions. For genomic selection to be worthwhile, we really need it to be more accurate, and it needs to be cheaper. And really starting to move more towards prediction-based selection, not necessarily based on genomics or just based on pedigree, but really based on many different kinds of data, including high throughput data. And it's really important that we increase genetic gain in terms of time and cost, because cost is extremely important for increasing the selection intensity. If we cannot have very large populations, then we can't increase genetic gain. So far, just a little bit more conclusion. So far, prediction-based selection looks like it's most advantageous at the moment for yield, but we really have a lot more work to do for other traits, including rust resistance. With that said, increasing genetic gain for yield is an extremely important component of getting rust resistance out to farmers. So it's even though that we're not quite there yet with rust, we'll still see the benefit. So with that, I'd just like to acknowledge the sources of funding. I won't read off all of them for the sake of time, but we have a lot of different sources of funding. Most importantly would be the Durable Rust Resistance in Wheat Project. That's it. Thank you.